you a little bit and then you can start. Okay, Alina, is it okay if I start, or is everything ready for the
Yes, we are ready.
Yeah. Okay, great. Okay, so I'll just introduce you, James. Dr. Jim O'Donnell is a physical oceanographer. He earned a bachelor's in science degree honors in applied physics from Strathclyde University in Scotland, and a master in science and PhD in oceanography from the University of Delaware. After serving as a postdoctoral research associate in the Department of Applied Mathematics and Theoretical Physics at Cambridge University, England, he joined the faculty of the University of Connecticut in 1987. In 1999, he was promoted to the rank of professor and served as an interim head of the Department of Marine Sciences and director of the Marine Science and Technology Center from 2002 to 2005. He was elected to the Connecticut Academy of Science and Engineering in 2009 and was appointed to be Executive Director of the Connecticut Institute for Resilience and Climate Adaptation, CIRCA, in 2014. Professor O'Donnell's research focuses on understanding the physical processes that determine the circulation and transport of materials in the coastal ocean. With students and research associates, he is currently involved in both the construction and testing of models and the development of technology to make observations. He is also interested in fundamental geophysical and environmental fluid dynamics and the application of mathematical and statistical methods to the development of models of biochemical processes. His research team has sustained four buoy-based instruments, arrays, and 5 high-frequency radar surface current sensors in Long Island Sound for 20 years to provide data for both practical and science applications. As Executive Director of CIRCA, he leads a team charged with the creation and dissemination of practical and sustainable strategies to enhance the resilience of the built environment and natural ecosystems in Connecticut. Great. So I'll give you the word, the microphone. You can start, James.
Well, thanks. So I appreciate the invitation. It's an honor to be here, and I'm embarrassed by my inability to speak Spanish, but I hope you all understand my English. I grew up in Scotland, and I still retain a pretty strong accent, apparently. Though it's diminished a lot since I first came to the United States. I think some people still have a tough time understanding me on occasion. So if you have any questions, or if something is unclear, please just chime in and I'll try and say it again. And I'll try and speak slowly, but I'm often, I get excited about stuff and speak a little more quickly. So these are these two figures I'm showing here are the results. And I'm going to tell you about a problem in which I've used physical oceanography to help people who are biogeochemists and managers of water, responsible for water quality and ecosystem health, detect the effect of management actions. And so that's pretty vague, but I'll be more specific in a moment. And I'll come back to these figures, but these are pretty important. On the left side here, let me use my arrow. Can you see the arrow here okay? Or how about now?
I'm not sure. Try again. Yeah, I can see it. It's just tiny.
Okay, oh, it's tiny. Maybe
I see it.
Use this thing. This is how about the laser pen? Okay. That didn't work. Okay, it's an arrow again, I guess. So on the left here, I'm plotting on the horizontal axis the what's called the load, the wastewater treatment plant load in pounds of nitrogen per day from water treatment plants that are discharged into Long Island Sound. And over the last 30 years, all the towns in New York City, around Long Island Sound and New York City, have been required to reduce the amount of nitrogen they discharge into the sound, and they've done it. It's cost several billions of dollars to do that. But basically what's happened as the load has decreased, it's gone from like about 50,000 pounds per day down to 20,000 pounds per day. The area of the bottom of Long Island Sound that's hypoxic has reduced. And there's some error bars and lines here. And what I'm going to tell you about is why that has occurred. So we jump to some more geography. So on the left side here, we have a map of Connecticut, right? And to the south here is Long Island. It's in the state of New York. And the distance between this area over here where I'm pointing is the edge of New York City. And over here where I live in eastern Connecticut, it's about 200 kilometers. And the width of Long Island Sound here is about 40 kilometers. And then on this map, there's probably 4 or 5 million people. But to the west here, where New York City is, there's another 5 million people. And the water treatment plants that service all those people drain into these locations which are numbered. These are the sources of wastewater that discharges into this little this water body. And there's a little line here. It's called that's like a channel. It's called the East River, but it's not really a river. It's just a sea level canal. And seawater sloshes back and forth between western Long Island Sound, which is I'm pointing to here, and the New York Harbor, which is where New York City is, and fresh water and then the dissolved nutrients get transported from this area in the East River into Long Island Sound, where they promote phytoplankton growth in the summer. The turbidity is very high in Long Island Sound, so when phytoplankton get mixed out of the top meter, or they die, they begin to respire oxygen, and that reduces the oxygen concentration in the water. And so all these bar graphs show the nutrient discharge from different districts within Connecticut between 1995 and 2010. And what you see is they've all dropped roughly half. All the ones in Connecticut are pretty small, though, relative to these ones in the East River. There's area 9 and 10. The unit there is 50,000 pounds kilograms per day, where the ones over here are like 4,000 or 5,000 pounds. So these are all different scales. So the big source is around here where most of the people live. On the left here, you can see the same data, but in a time series. It starts about 1995. And each of these lines is the discharge of nitrogen in in pounds per day from different districts. So number 8 is this one over here by the East River. Number 9 is this one down here, for the furthest west. And you see they're much bigger than all the other ones. So in 1985, it was noticed that in this area of Long Island Sound, the western part of Long Island Sound over here, the bottom water became hypoxic in August, and it was pretty extensive, and I'll show you some maps in a moment. And then in 1988, there was an agreement established. It was really the US EPA, they enforce a law called the Clean Water Act. And the Clean Water Act requires that when the water body is deemed to be impaired, that means it's been negatively impacted by pollution, then the states have to act to rectify it pretty promptly. And so there was a lawsuit, and an agreement was established between the state of Connecticut, the state of New York, and New York City, and that they would all reduce the nutrient loads to Long Island Sound. But it takes many years to organize the money and develop a design to change water treatment plants. These water treatment plants are very expensive, and they're in areas which are highly developed, so there's not a lot of space to modify them, and then they have to do construction, which is a big project. So but between 1985 and 95, the money was raised and projects started. You can see that these two drops occurred in the nutrient loads from the two biggest water treatment plants. And then number 8, which is this area over here, it's called, the plant is called Newtown Creek. And it began to creep up. And then in 2003 or 4, another project was initiated. And then in 2006, when it started working, the nutrient load dropped a lot. So these sort of 3 periods of high, medium, and then since 2015, it's been low nutrient discharge. These have a response in Long Island Sound. We expected to reduce the area of hypoxia. So hypoxia is defined in Connecticut and New York to be less than 2 milligrams per liter. So every month and then every 2 weeks in the summer since 1988, ship surveys have been executed to which the ship goes and visits all these dots locations, and they do a vertical profile of salinity, temperature, and dissolved oxygen, and they take nutrient concentration samples with bottles, and they do quality control, and they send all the data to a database which my team operates, and we share it now. And what I'm showing here is a map of the bottom dissolved oxygen concentration in August of 2012. And this is probably one of the most extensive areas in the 30-year record. You see, the red is between 1 and 2 milligrams per liter, and at that concentration, fish and organisms that live in the sediments frequently die. And the orange is 2 to 3, and those concentration ranges, it's thought that they are impaired. And then 4 and 5 are sort of 3 to 5, which is the yellow-green colors. Those are noticeably impacted, but it's this area of red that's really concerned. But the law that was enacted requires that the area that's less than 3 milligrams per liter, which is the orange and red, it has to be reduced by about by 70%. That was the goal. So every year since it started, people have looked at the areas of the hypoxia to see whether or not this investment of money has been working. And I've been at UCONN since 1987, and I started looking at it about 1993. And I wasn't really involved in the program development, but I was integrated into about in the early 90s, and I started looking at the data. So, and it turns out that detecting whether or not there has been a change in the area of hypoxia is not simple, and I'll show you for some reasons why. And the first reason is that in the early years, there were fewer samples. In 2012, there was probably 30 stations occupied every 2 weeks in the summer during this period of hypoxia, and then every 4 weeks during the winter. And there's 2 ship surveys that are separate, but at the same time. There's the one that goes in the Western Sound. These are the black circles, and then the small circles are a different ship. But in the early years, like in the early 80s, like 86, 87, 88, there was only one ship, and they only did 5 stations. So if you do 5 or 6 stations in the middle of the Sound, your ability to estimate the area of hypoxia is pretty limited. Right? That you have to assume that the station, say F3, is representative of the whole area across the Sound. And so that introduces an uncertainty. So the early cruises have to have a large uncertainty relative to the later cruises. So that's one problem. So I could evaluate the magnitude of that error by taking data from a later year, say this is August 2005, and then just comparing the area of hypoxia identified using the extensive surveys by just subsampling it, pretending that I only had these 4 or 5 stations, and calculating the area of hypoxia, that is the area less than 2 or 3, from this set of data and from that set of data, and comparing them. And I could do that for lots of years, because since the mid, say 93 or so, we've had much more extensive data available. So that's one thing I did. I computed the effect of what I call the array bias. That is the bias in the estimate and the uncertainty in the estimate associated with the sparse resolution. So this is an example that was a graph I made in 2020, which shows the area of hypoxia in square miles is basically every August since 1988. In the first few years here, which I've labeled with this little red parenthesis, and they were conducted by a lady who was a professor here at UCONN when I first came here, Barbara Welch, and she only was able to do 5 or 6 stations on her cruises. And so these ones in the early years are very uncertain. These later ones are higher quality. And so what people have been doing, so every year there was another bar was added to this, and every year people would draw a line through it and say, well, is it increasing or decreasing, or are we on track? And that's a complicated question. So the people who were managers and who were responsible for the program, they always said it was decreasing, and it does kind of look like that. But if you look at the difference between the data, which is the bars, and the red line here, this is this figure on the top right is what you get. That's the residuals in the fit. And so if there was a linear time trend in the area of hypoxia, then these residuals should be normally distributed, and they should not be correlated. So that means like each one should be independent, and there shouldn't be like a whole bunch of positives and then a bunch of negatives, right? That's indication of serial correlation. And so you can evaluate whether they're normal, the residuals are normal or not, using this diagram, which is called a QQ plot. Basically, it's a value of the residual plotted against the probability of the residual if it was normal. And what you see is a whole bunch of points here at the extremes, which are off the line, and therefore this is strong evidence that the residuals are not normal. And there's correlation. You can see that there's five points here which are all positive, and then 5 here which are all negative. And so you shouldn't believe any linear regression in which the residuals are not normal and for which there is serial correlation. So I don't think that, so I was asserting that there's no evidence of a linear decrease with time with this data. However, I wanted to try and figure out whether these differences between the estimate, the linear line, and the real data were due to some physical process, or was it just due to the fact that the way that the area was estimated was uncertain. And I'm going to tell you about how you can estimate what the uncertainty in an area estimate is. So here's a map of Long Island Sound. The blue color here is bathymetry. And at these locations here, Execution Rocks and Western Long Island Sound, I've maintained buoys since about 2004, so 20 years now. And these buoys measure lots of things, including dissolved oxygen near the bottom. And then in about 2012, I got money for another one called RG. There's also another one out here in the Central Sound, but it's for a different purpose. So all three of these have records of dissolved oxygen that are obtained every 15 minutes. Now, these are, and this is the data from them. And what you see here is a typical, this is actually from 2017, but there's 20 years of these, and they all look kind of the same. What you see in green is the bottom value at Exeter Rocks, which is this station here, the furthest west. So, and we turned it off in mid-August in 2017 for some reason. And the blue line here is ArcG, that is this one over here. And the purple one is the middle, that's the Western Sound Buoy, or WLIS. And they all show the same characteristics. Like in June, the concentration of dissolved oxygen is quite high. And then by mid-August, it's quite low. And here's 2, that's when things die. And 3 is when it's become hypoxic for the purposes of the agreement with New York City and New York State. And both the Western Sound and Exeter Rocks are hypoxic by the beginning of August, and that's typical. Thing that I noticed though, in the first year, after the first year we had these buoys in place in 2004, I noticed that there were intervals when the oxygen concentration at the bottom goes up. It goes up rapidly, actually. So it falls. The overall trend is basically decreasing through August, but it's interrupted by these periods when the oxygen jumps up. And so in this particular period, it jumped up to 5, right? Almost doubles. And then here it's about 1.5, and it jumps up to 4 for a period of like 10 days. And then it drops rapidly again, and then this sort of slow trend continues. So the reason that I'm pointing this out is that if you go out with a ship every 2 weeks and sample something like this, there's a good chance that you would miss these periods where what I call ventilation occurs. By ventilation, I mean oxygen is brought to the low oxygen areas of near the bottom. And during each summer, there is 2 or 3 or 4 of these events where for a week or so, the oxygen level is increasing and gets high, and then followed by this rapid decrease again. And that puzzled me. And I wrote a paper with some colleagues in 2008 called Intermittent Ventilation of Long Island Sound, that kind of explains or poses an explanation for why these oscillations or intermittent ventilation events occur. But for the point of this discussion, what's important is that the ship surveys alias these events, and so each sample you take with a CTD from a ship is not really very representative of a 2-week average, right? It's just or if you want it to be a representation of the 2-week average, you have to apply a sort of large uncertainty to that. And so that's what I did. If you draw sort of a line through this stuff, or a black line is a low pass filtered series with a cutoff of about 2 weeks. So it's like a running average, roughly. And then then I pick the middle value of this low pass filtered data in every 2-week intervals, and I compare that. And I calculate the standard deviation about that. And if you calculate the standard deviation of the low pass filtered records for each of these buoys, and you do that for 20 years of data, you can estimate what the standard deviation is at each station during the summer. So I did it for August. And at the Exeter Rocks buoy, the variation or the uncertainty that should be applied to the concentration estimates from ship surveys is about 0.6 milligrams per liter. And at the Western Sound buoy, it's 0.3 milligrams per liter. And at the ArcG buoy, it's about 1.75 milligrams per liter. So the way you should think about that is that if you want to make a map of the area of hypoxia, and you've got like 30 points, each of those points has to be treated as if it's an estimate with an uncertainty, and the uncertainty estimate is spatially variable. And so to make a map, then I assumed that the west of Exeter Rocks, the uncertainty was about 0.6, and east of ArcG it was about 1.75, and that leads to black line shows my assumed spatial structure of the uncertainty that I would apply to each of the ship survey data points for the near bottom dissolved duck stream. And these are, so then what I did, sorry, let me move it back a little bit. So then to create a map like this, I used the actual, this is actually based on the real data. So at station 19 in August 5th, in August 2005, at station 19, the value of the concentration measured was about 3.5. And at E1, it's about 2.5 or something, right? But it could have been 3.5 plus or minus, like, plus or minus, where is it? This value, plus or minus 0.175, right? Because if I'd done it again and there was some, if you treat those, the uncertainty as a random variable, any value between the value you got plus or minus 0.175 is plausible. So what I did is I made this, sorry, let me get back to the map. I made this map. This is the real data. Then I did it again, but I added a random number multiplied by the standard deviation estimated at each station location and made another map. And I calculated the area again. I did that 1,000 times. So I can't, and to put it succinctly, what I did is I Monte Carlo simulations of the effect of noise on the estimate of the area in order to calculate the uncertainty in the area attributable to the noise that's not resolved by the ship surveys. Is that clear? Has anyone got any questions about that before I go on? Okay.
It's clear for me, yeah.
Sorry, I didn't hear that?
Yeah, that it was clear for me.
Okay, good. So here, I then, so for each cruise then, I can calculate the area, the hypoxic area, and I plot that here in the horizontal axis, and it's kilometers squared. And the orange is the area of the sound, which is less than 3 milligrams per liter, right? And I plot here the 68th percentile of the uncertainty. Sorry, this on the vertical axis is the uncertainty in the area estimate. And it says 68th percentile, or two standard plus or minus a standard deviation. So, for example, this point here, I'm pointing to the area of hypoxia from that cruise was about 600 kilometers squared. But after I'd done the Monte Carlo simulation of the effect of the errors on that, I get an uncertainty of about 175 kilometers squared, right? So that would be about 30% of the value of the area estimate. Now, obviously, as the area expands to the east, right, the area gets bigger, but the uncertainty doesn't get much bigger though, because there's more points being included in it, and that sort of smooths the estimates a little bit. So most of the points seem to lie along this black line. There's 4 or 5, 6 cruises in which the uncertainty estimate is much bigger. And several of those are 91, 92, 93, right? And those are partly a consequence of the limited sampling that was available on those early cruises, right? And so roughly for each cruise, you can do the Monte Carlo simulation and calculate sort of a precise estimate of the uncertainty, or you could just take the area and use this black line as an empirical estimate of the uncertainty in the area. So now I want to revisit this question of, is there a trend in the data? And so to do that, taking into account the uncertainty, I'm going to use a weighted least squares to fit the trend line. What I showed you originally was what's called ordinary least squares, and it just minimizes the difference between the model and the data squared. And the model would be like a constant times a coefficient times time, right? That would be a linear trend model fitted to data with the ordinary least squares. A weighted least squares does a similar thing. It minimizes chi-squared, which is a difference between the model and the data squared, but it's normalized by the uncertainty in the data. So that points in which the uncertainty in the data is big are weighted less than in the points which are more precise, right? And then the nice thing about that is that we can estimate how well the model should be expected to fit the data. So for example, and this is an important point, that if the model was perfect, if in fact the area was reducing linearly with time, but there was noise in the data, you shouldn't expect the correlation between the data and the model to be 1, right? It has to be less than 1 because there's noise in the data, and you can estimate what the expected chi-squared should be if the model and the data and the noise that you assume are consistent. And roughly, for large degrees of freedom, that is for lots of samples, and we've got for each cruise, for the cruises, we've got roughly 30 points now. That's quite big. So we should expect that the weighted least squares minimum chi-squared should be about 30. Right? Now, if it's much bigger than 30, then the model is not a good fit to the data. And if it's much smaller than 30, then the errors have been estimated to be too big. So here's the result, what you get. Again, the data here, or the blue points are the data measured. They're the best estimates of the area estimated from the ship surveys. And then I put on those the standard deviations, plus or minus the standard deviation of the area estimate based on the Monte Carlo simulation. You see here first that the early ones are big errors, right? And so they can't be trusted as much. Whereas the later ones, there's more stations, and those errors, those points are better. So the red line shows the ordinary least squares fit, right? And then the green line shows the weighted least squares fit. And you see it fits better up here where the uncertainty is smaller. And you can see that line actually, it passes through most of the data points. It passes, all the lines pass through the data points here when the area is pretty uncertain. And that's a well-known problem, that any model will fit noisy data. These data points are not very valuable because the uncertainty is large and they don't discriminate between good models and bad models. When the uncertainty is smaller, then the model, only the blue and green lines are consistent with those. The red line doesn't go through these points, for example. But because these errors are big, we weight those points less, and so these two lines go through there. Now, the reason that there's two lines is that there are obviously some anomalies here. There's an anomaly here, even though the error, the uncertainty is small. This point is not consistent with this green and blue point, and these points aren't either, and neither is this point. So there are 5 points here. This one in 2014, this one in 2012, this one in like 2003 or 4, and this one. Those are inconsistent with the line, but overall, you know, this blue and green is a better estimate. And I can look at the chi-squared for each of those, which is the misfit. It's like a residual. Again, you can see these points where the peaks are. They're inconsistent. And if you calculate the total chi-squared, it's larger than 30, and it's not a good fit. Now, there's no reason, though. After I'd done that, I realized, you know, for the last 15 to 20 years, I had been assuming that the trends should be linear in time. And there's no reason to assume that, because I showed you earlier that the nitrogen reduction had not occurred in a linear fashion. It had occurred in several steps. So instead of plotting using weighted least squares through the points which are in the lower cluster, both the black line actually show this black line here. The black dashed line is all the data. And if you exclude this point, the 1997 point, you get the red line, and then the purple line, and then if you exclude the top one, you get the red line. So there are 5 years over this 30-year period which are inconsistent with the fact that it's inconsistent with the model that the area of hypoxia is linearly dependent upon the annual average waste load. So now this red line, this is what was predicted by people who designed the update to the water treatment plants. They thought if you invested enough money to reduce the waste load from like 50 to 20, that the area of hypoxia would go down to about 200 kilometers squared. Which is, and when I plotted this, I was pretty surprised that most years are consistent with that expectation, right? That you can see, if you didn't look at the top 5 and this bottom one in 1997, you would believe that the model was correct. And if you look at this thing over here shows the QQ plot, it's called. It's the error in the fit against the probability of that error, and you expect the points all to lie on this line. And most of them do, except these ones which I've labeled 2012, 2003, 94, 2018, and 2015, and I've highlighted those in this graph here. They're clearly inconsistent with the model. These error bars are not that big, but they're several times the error with the model. So jump one more. So this is the way I interpret this, and I think it's pretty robust, is that the investments that we've made in water treatment plant upgrades over the last 30 years have basically worked. You know, there are 25 out of 30 years, 25 of them are consistent with the expectation that the nutrient load controls the area of hypoxia, and reducing it causes the area of hypoxia to shrink. And in the recent years, the area is so small, it's going to be hard to detect further changes. Like the uncertainty in the area now is roughly about 50 kilometers squared, and the area of hypoxia is like 200 kilometers squared. So further upgrades might reduce it by 50 or 100, but that's going to be hard to detect with an array which is built like this. We have to change the station location to resolve smaller changes that may occur in the future. But the big question is, why are there 5 years that are anomalous? And I don't know. For the last year, I've spent some time trying to look at records to figure out whether or not the weather was anomalous, or something about the discharge was anomalous. And in one year, I think it was 2003, there was a power outage in New York City for a few days, which shut down the water treatment plant, or some of the water treatment plants. And so the amount of nitrogen that was getting discharged into Long Island Sound is probably much higher that week and it was in August. So the discharge may have been anomalously higher than it would have been, so in which case this area to be consistent with the model, this point would have to be sort of way over here, which would be maybe 2 or 3 times the nutrient discharge for the year. So that's hard to believe. And I've looked at temperature and salinity and river flow and wind and things, and I haven't got a smoking gun. But one thing I do know is that in these 4 years, that ones that are very high, the area, I mean, looks a lot like this one, where oxygen concentration along the north side of Longland Sound is higher than normal, and it's asymmetric. So there may be some physical mechanism which causes the low oxygen water to move to the east along the north side of Longland Sound. I don't know why, but there might be something, and that's kind of what I'm looking into just now with models. And so let me just, I did one other thing that's sort of interesting is I built a little software tool that allows, that runs on a web services that accesses the data which we get from the ship surveys through an AirDAP server. And then it does the calculation to make the area map and creates uncertainty estimates too. And I also built, it also calculates the volume of the water that's less than a threshold of 3 and 4 and 5, but 3 is the one that we're most concerned about. So in addition to calculating the area of the bottom, which is less than 3, calculate the volume of water that's less than 3. And I thought that that might reduce the uncertainty. So for example, it could be that some years there's a really thin layer of hypoxic water that spreads further afield, and so the area gets bigger, right? That was a possibility. So I computed the volume, and it basically parallels the area. It doesn't, the layer thickness of the water that's hypoxic, it tends to be about almost 10 meters, and it doesn't really change from year to year. So here's my conclusions. I've developed a method to estimate the uncertainty that combines data from buoys and ship surveys. And I've showed that there's a linear dependence of the area on nutrient discharge, and that is statistically significant. It's absolutely consistent if you omit the 4 or 5 years when the nitrogen load doesn't explain it. And I've got here my comment about there was a hypothesis that I sort of started years ago in the paper in 2008, and others sort of followed on with that there is a effect of wind that might cause hypoxic area to be bigger. But I tried that in a regression too, in a rigid way, and it didn't work. So I don't think wind explains it. Finally, with the current loads, we should expect that in 80% of the years going forward, that's 4 out of 5 years going forward, the criteria that was established for the acceptable amount of hypoxic area will be met. Is the 2 milligrams per liter area and the 3 milligrams per liter area will be less than what we're expecting. But there are 1 out of 5 years will be bad and the area will be bigger. Now, one thing that's interesting that I didn't point out was that this red line is the slope of the line using weighted least squares through all these data points. And this line here is the weighted least squares fit through these 4 points, and they're parallel, right? So even in the bad years, the reduction in the hypoxic area has worked. You see, this is 94, 2003, 2012, and 2016. And that's just where nutrients are getting smaller and the area gets smaller at the same rate it does during these other years. So it's just kind of curious and interesting. Maybe there's something to learn about that. So all in all, it's a good story. That is the Clean Water Act, which was passed about 40 years ago, implemented 30 years ago, has worked, and it only cost $3 or $4 billion. Any questions?
Thanks so much, James. That was really interesting. Does anyone have a question? Yeah, I see Alex already has a question. You can open your microphone and camera if you want, Alex, and ask.
Okay. Well, hi, James. Thank you so much for the presentation. So I just have a question. Do they take samples from the sediment during these surveys, or it was just water samples that you performed?
Yeah, well, so I mean, that I know where you're going. Yeah, they don't take sediment samples, but there's a lot of other work that has been going on to try and understand why hypoxia occurs, and what is driving it. And there's a big effort being put into development of a numerical model to try and understand the variability between the good years and the bad years, etc. And I'm sort of involved in that. And as part of that, we've developed instruments to measure the respiration rate in the water column and on the bed. And also, I'm working on a project to use oxygen isotope ratios to try and discriminate between the respiration that's occurring in the water and respiration that's occurring in the sediment. But basically, when the hypoxia is bad, it's down below 2 or 3, the effect of the sediment respiration is very small. Most of the respiration occurs in the water column.
Okay. Oh, thank you. Yeah.
Any other questions? Someone wants to open their microphone and ask? If not, in the meantime, I can ask you, James. So you're going to continue the effort of monitoring water quality like this, or I mean, is there enough funding? Is the state paying for it, or because I mean,
yes.
yeah.
So yeah, the EPA, US EPA pays for it, and the buoy program is in jeopardy this year, temporarily. The buoy program has been going for 20 years, and it's supported mostly by NOAA. But the president's budget this year he's trying to show he's being frugal, and there's budget cuts that have affected us. But it's likely that our senators and congressmen will restore it, because this is a pretty well-supported initiative. And the ship surveys are very robustly supported by EPA, so it's going to continue. And because, this sounds like a big deal, right? The ship surveys every month for 30 years, right? It costs about $1 million a year for that. And it costs about $500,000 for the buoy up program. And so then on top of that, there's probably another $1 million that are spent in science upgrades and model development, etc. So, but you have to compare that to what was spent to upgrade water treatment plants, okay, and to operate a water treatment plant. Like and there's maybe 20 million people whose effluent goes into Long Island Sound. So that's a lot of people. And I think we spent between $2 and $3 billion over 30 years to upgrade water treatment plants. And so spending about 0.01% of that to see if it works.
It makes sense.
Doesn't make a lot. It makes a lot of sense, doesn't it?
Yeah, it does, definitely. I mean, it's just really nice to see that you have that sort of data set, and then you can continue to monitor and learn stuff.
Yeah. It's been. I've been at UCONN for 37 years, and so in 20 of it, I've been supported to develop these buoys and operate them. So I have a little team of people who've been working on it, and then and we can collaborate with other people too. So there are other people at UCONN now who I'm working with on this oxygen isotope analysis. And I've developed a little model about how you use oxygen isotope ratios to get estimates of the relative importance of benthic and water column respiration.
Nice. Are there any other questions somewhere, or otherwise I can ask you? I'm just curious about those years when those 5 outliers, let's say.
Yeah.
Was there any obvious impact in the ecosystems? Like, did biologists say, or something happened? Or, I mean, was there, an obvious?
No, the biology is hard to measure, right? That's the difficult thing is that, dissolved oxygen is affected by biology, obviously, right? It's a difference between the production rate and the respiration rate and the mixing rate. Right, so there's mixing from the surface, respiration in the water column and in bed, and then production in the surface. So those things are hard to measure, and and no one I've showed this to has produced any plausible biological correlate yet.
Okay. Yeah, no, I was just curious because I mean they're obviously much higher than any other year, so I thought maybe.
Yeah, the area of hypoxia is much bigger,
It's
so
much
the impacts
bigger, yeah.
you'd think would be more extensive.
Mm-hmm.
But and this is makes up the point I made earlier with this figure that when the uncertainty in the data is really big, right on the left there, any model fits. And so getting and getting data from biological processes always has big uncertainties, and so it's hard to discriminate any real inconsistency.
Yeah. Okay. Any other questions? Someone? I don't see any hands, but maybe anyone can open their microphone if they want to ask. Or do we have any questions in Facebook or Karina?
No, we don't have questions.
Okay. Well,
Well,
yeah,
thank you all for listening.
Yeah, thank you so much. It was really interesting, James. Maybe we can have you again talking about the other sea level and the impact on the coast.
Yeah, I mean, my work has sort of switched from being sort of fundamental to being applied physical oceanography, right? Mm-hmm. So this question was sort of an important question for public policy making. I mean, there is whether should New York City be required to invest further in upgrades of water treatment plants, right? And that was sort of drove it. And at the moment, it seems to me that they've met what was the goal was set 30 years ago. So the question now is does a new goal have to be met? And so I answered that question using physical oceanography techniques,
Yeah.
And the same is true with the climate adaptation stuff. I've been using methods I learned because I'm studying oceanography to help answer questions about what towns should do to try and limit the effects of climate change on their infrastructure.
Yeah, well, yeah, it's really interesting, really nice to see the applied science, and yeah, it's good. I mean, it's great that you met that they met this initiative to reduce by half the area and stuff. But then you wonder, is that good enough? Like, it's 50 square kilometers small enough, or should we like improve it even more? Or
well, I think that's exactly right.
Mm-hmm.
The area of hypoxia now is more concentrated in New York. So the Long Island Sound is administered partly by Connecticut and partly by New York. Mm-hmm. And so the Connecticut part of it now meets standards, but the New York part doesn't.
Okay.
So they may have to do more.
Yeah, exactly. Okay, well, many thanks, James. It was lovely to meet you and to have you as a guest. And yeah, we'll be in touch.
Thank you very much. It's great to do it. Bye-bye.
Bye.
Bye-bye. Thank you so much.